There we go. Right. So links first. Right. So hello, everyone. Welcome to session 7 of this year's BGA. And first off this week, we're going to have ModDotPlot by Alex Sweeten. You may have noticed I've just posted some links in the chat. The first one is a form that I'd like everyone to fill out if possible. This is just so I can make improvements to the course as we go along. And the code for it is .plot, so I can separate it all down by session. Next is the Gitpod link, which you will need for the Gitpod link in a bit, as well as the Discord and the YouTube links as well for everyone else. But yeah, hopefully we will have fun. So take it away, Alex.
All right, thank you, Damon. I sent a request to share screen. Perfect.
Yep, sorry about that.
All good. All right, everyone. My name is Alex Sweeten. I'm a third-year PhD student at Johns Hopkins University, and I'm also a research fellow working with Adam Phillippe's group over at the NHGRI. And today I'm excited to explore some satellite DNA with you using ModDotPlot. So before I get started with my presentation, I think I want to what am I saying? Go to the Gitpod and make sure that everything works, and also run a sample command while I give the presentation, just so we save some time because we only have an hour today, and also to make sure everything works the way that it should. So if you're going to the GitHub page, make your way over to docs/tutorial markdown file, and launch the Gitpod. When you launch the Gitpod, you should get should take maybe 2 minutes or so. It's going to download some files, download all the external dependencies for ModDotPlot, and it will give you this nice message ready to make some dot plots when it's ready to go. So once you have that ready to go, copy this starting command here. That's going to be this generating a basic plot. You can go ahead and copy and paste it into your Gitpod. And if all goes well, it should showcase the logo as well as a progress bar. And so while the progress bar is going on, this command should take 10 minutes to run. I'm going to go over my presentation. And the presentation, I'm hoping, should take around half an hour. In that case, afterwards, we can go ahead and play around with the rest of the tutorial that I'm showing here. Okay, so moving on to the presentation and some motivation for you. Around 2 years ago, the Telomere to Telomere Consortium published a landmark paper that they had finally completed the human genome. This paper was published in Science and was the result of years of work and really an improvement both in assembly algorithms and sequencing technology. The overwhelming result of this is that the regions that are new to this reference genome compared to what we couldn't see before is that these are pretty much all repeats. The overwhelming majority are satellite and segmentally duplicated sequences. So SenSat regions, these are satellites that are apparent in the centromeres, make up almost 3/4 or 150 megabases of new sequence. Segmental duplications, these are just large duplicated blocks, make up a little over 1/4. rDNA, which is a very repetitive gene array present in the acrocentric chromosomes, make up almost 10% of this. So just a lot of repeats are what have been added to the reference genome here. And so satellite DNA is maybe a term you're not too familiar with, just because in reference genomes, there hasn't been large satellites that we've been able to see before. So you might not know what it is. So simply put, satellite DNA consists of tandemly repeating sequences. The reason why it's called satellite DNA is because back when you would do buoyant density centrifugation, I'm showcasing it here in this little cartoon test tube. You would get separate satellite bands compared to bulk DNA, where the bulk DNA is in black, and then the satellites are these little colored bands here. And this is due to the fact that satellite DNA has a very repetitive structure and different GC content that makes it appear differently in this situation. So satellite DNA varies pretty dramatically, both in terms of the size of the repeat, that's the size of the arrow here. These can range from as little as 2 base pairs, like a simple AT repeat, to very large 1,000 base pair repeats. And the array size, the length of the total number of arrows, also varies pretty dramatically. You can have mini satellites, which are very small, under 100 base pairs or so, towards huge macro satellites that are mega base pairs in size. And so these satellites are quite interesting, both because they play a lot of roles in diseases and things, but they also differ just in terms of GC content and are very variable between humans. So satellites, okay, they're cool, they exist, but who cares about them? Why should we care about satellites? We've gone 20 years with a reference human genome without satellites, so why do we need them now? What's important about them? And if you don't have satellites, well, you die, basically. Satellites are essential for eukaryotic life, specifically for humans and also for great apes. A special type of satellite called an alpha satellite is required for kinetochore binding. So for cell replication, this is absolutely necessary. So you may have been to my colleague Sergey Korin's talk on Virco last week. And he sort of has this mantra in our lab, which is that if I synthesize an assembled genome, do I get the same living organism back? And without those centromeres, instead of getting this nice live plant, we would get a dead one because we can't function without centromeres. And just to clarify that plants don't require alpha satellites, but they still do require satellite DNA, different kind, in order to facilitate the kinetochore binding during replication. So in addition to centromeres, off-centromere satellites also are showcased in human diseases. This was a very cool preprint that came out around a month ago by some members in our lab looking at the factor of Robertsonian translocations. This is a structural rearrangement present in around 1 in 800 humans. So in the short arms of your acrocentric chromosomes, you have these areas called pseudohomologous regions. And these regions tend to pair up during meiosis, recombine, exchange material, and then segregate. So what happens here is that in chromosome 14, this area is inverted with respect to chromosome 13 and 21. So as a result, when these pair up in the pseudohomologous regions, sometimes they can pair up in an inverted manner. What this leads to is that instead of the normal gene pairing up where you have short arm to short arm, long arm to long arm, you can have you can mix and match. So the end result here is that you have a chromosome fusion of either 13 or 14 or 14 and 21. And while this can be phenotypically normal for the carrier, for any progeny, this can lead to things like trisomies or increased cancer risks. So this is a very prevalent observation. And this is all mediated by this SST1 satellite present in the pseudohomologous region. Some other repeats that are worth caring about. We have tandem gene arrays. These don't necessarily fit the classification of a satellite because they are genes, but they are tandemly repeating, and thus we can see them with ModDotPlot, so they are worth talking about. Some examples are the TSPY gene array in chromosome Y. This is responsible for certain male sex characteristics, as well as rDNA in the acrocentric chromosomes. So this is chromosome 13, 14, 15, 21, and 22 in humans. This is necessary for life. We need rDNA to get our RNA. Some other repeats worth caring about are palindromes. These are a type of inverted repeat. So you have some forward repeats and then some inverted. These are also quite prevalent. We also see some large duplications and large inversions. These are not necessarily tandem repeats because they only occur once. You can also think of these as low copy duplications or inversions, but they do play a pretty significant structural effect, especially considering that around 25% of the newly sequenced DNA from the complete human reference genome contains these large duplications. Okay, so hopefully I've convinced you that repeats in satellite DNA is worth caring about. So how can we visualize them? And spoiler alert, it's going to be with a dot plot. So this is the traditional definition of a dot plot. It's a matrix consisting of two sequences. You have sequence X on the X-axis and sequence Y on the Y-axis. And whenever you see a match of a certain length, in this example, the match's length is 4, we draw a line there to show that there's sequence similarity between there. And so you can have some flexibility with how you design your dot plot. If you have the same sequence on the X-axis and the Y-axis, this shows self-similarity, so where in the sequence things can repeat. This can also show orthologous relationships if you have different sequences on the different axes, which is useful for, say, comparing gene regions or seeing if there's any structural variations between one organism to another. And so this works pretty well. This has worked pretty well for decades. This is considered a vectorized approach because you're drawing lines with matches. However, for larger satellite repeats, this vectorized approach really loses its applicability. So here what I'm showing is one satellite repeat on the X-axis, one on the Y-axis, and the satellite repeat is a simple variance of the human satellite 3, GATTG. And you can't really make a whole lot out of this dot plot. It's very overwhelming and confusing. It's also, not to mention, super computationally intensive to do. And this is just for 400 base pairs, when we know that satellites can span mega base pairs in size. So this vectorized approach needs some updating in order to work for our purposes, which is to visualize large satellites. And so around 2 years ago, this paradigm shift in terms of dot plots came into effect through a publication by Mitchell Folger from the University of Washington called Stained Glass. And this converts the vectorized model of a dot plot into a rasterized model. So what I mean by rasterized is that when you're not drawing lines, but you're instead fixing at certain intervals a discrete value. So if you see this dot plot on the right here, each pixel represents a pair of intervals based on some fixed size. So in this example, we have a fixed size of 1,000 base pairs. And so this highlighted pixel here that's teal in color represents the region from 500,000 to 501,000 to 800,000 to 801,000. And we can see, based on our mapping from our sequence identity to the color, that this is around 85% identical. And this is nice because we can compute sequence identity for each pairwise combination of intervals. This adds another layer of information to our dot plot that we wouldn't get by just a standard vector. We can see exactly how similar these two regions of the genome are to each other. So how do we compute sequence identity? Stained Glass uses Minimap. I'm sure most people have heard of Minimap, super popular read mapping algorithm. And it estimates identity by combing through each pairwise combination of windows and computing the number of matches, mismatches, insertions, and deletions, and plugging it into this formula to get an estimate of the identity. So this works, but it scales quadratically with the genome size. If you have a genome of size n, you're producing a matrix of size n squared, and you're doing computations on n squared values. So this quadratic scaling is quite time intensive and leads to high computational overhead, so we can do better. We can save time specifically by sketching within our intervals. So if you're unfamiliar with the idea of sketching, you can think of it as taking a smart subsample of whatever data point that you're looking with. So in this example here, we have the Mona Lisa. what properties can I remove from this painting that will reduce the amount of information we have, but still make it recognizable as the Mona Lisa. That's the basic idea behind sketching, that you can take some data away from it, but it also has a guarantee that certain properties are retained here. And so for our case, we're going to use the Modomizer sketch. So the Modomizer sketch is quite simple. So first, you convert each pairwise combination of intervals into sets of k-mers. And then given each k-mer, you plug it through a hash function so that you get an integer. And then you divide that integer by some value s, and you check if the mod s of that value is 0. So if it's divisible, for example, by 2, then you can just check if it's divisible by 2 by if it has a 0 in its least significant digit. And if it is, then we put that k-mer into our sketch. If it's not, then we can toss it away. We no longer need it. And so this is a sketching approach, and the property that it guarantees is that it guarantees a hierarchy of k-mers. So what I mean by that is that if you divide all of your k-mers by 4, well, all of those k-mers are also divisible by 2, just by nature that anything divisible by 4 is divisible by 2. We can determine that by looking at the least significant digits. Everything that has 2 zeros in the least significant area also has 1 0 in its least significant area. So this is a very efficient way to sketch our sets. And it also has this nice hierarchical property where we are guaranteed that certain k-mers have to be in certain sets. And so we can exploit this property of hierarchical monomizers to construct dynamically changing dot plots. So in this example here, I have 3 different dot plots. All of them use the same number of sketched k-mers. That's this monomizer section here. So when we have a large field of view, like we're looking at an entire chromosome, we can be pretty aggressive in filtering out our k-mers so that we don't have a lot of overhead when we compute a large dot plot. As we zoom in, we can be a little more lax with how many monomizers get into our sets. And if we zoom in really hard, we can pretty much keep every k-mer because it's a very small field of view. So computing it is not a big deal. So this is the approach that we use for ModDotPlot, and it works really well. So compared to stained glass, we see pretty significant improvements in runtime. So this bar chart on the right compares the runtime of stained glass to ModDotPlot for computing the distances of the Arabidopsis genome. This is a 125 megabase per genome. And ModDotPlot can estimate identity in around 5 minutes compared to stained glass, which takes around 6 hours. And you might think that, oh, okay, with that decrease in runtime, and because we are estimating with sketching, we're going to lose a lot of data, so it's not going to be as accurate of a dot plot, but we actually find that's not true. This is a comparison of 2 centromeres of Arabidopsis, and we can see that their profile of the dot plot is pretty much the same. It's very hard to distinguish any differences between these 2 plots. Only that one was computed by stained glass and took 6 hours, and the other was computed by ModDotPlot and took only 5 minutes. Okay, cool. We have a fast tool to build dot plots. Now what? So you don't have to be a divination professor in order to interpret the results of your dot plot. They are pretty straightforward, and it is very useful to know exactly what you're looking for. So if you just run, first of all, we're going to look just at self-identity plots. So we're not comparing to another sequence. We're just taking one sequence and comparing it against itself. So in that case, ModDotPlot is going to output 3 main files. It's going to output a full matrix. This is a same sequence on the X and Y axis where you have the diagonal. This is the same sequence matching to itself. So it's going to be very strong identity. And then any off-diagonal elements are going to represent repeats. You have a triangle. If you notice that this with a matrix, you have the upper triangle and the lower triangle, and they both relay the same information. So you can just chop the top triangle off and use that as your information so that you're not dealing with any repetitive data. And it also looks cleaner for publications and whatever application you're using. And when I refer to the diagonal, when ModDotPlot is laid out in this format, I am talking about the line on the bottom. So even though it's not diagonal, that's what I'm referring to. And then finally, it will output a histogram. So this histogram is useful for comparing identity thresholds. So for example, what does red mean? Red means you have very high identity estimate of around 100% compared to purple, where you have around 86%. And it's also useful for seeing how much of your plot reigns into a certain identity pyramid. Okay, so now moving on to how do we interpret these things? Anytime you see a triangle on the diagonal, that represents the set of satellite DNA. So remember that satellite DNA is tandemly repeating in this tip to tail fashion. This is organized in a dot plot as a triangle. So anytime you see a triangle on the diagonal, that's a satellite sequence. And you can see that in this plot, we have two alternating satellite classes. We have human satellite 3 shown in this yellow and a little bit of red triangle. And then we also have alpha satellite, which is present here. So sometimes when you're doing a dot plot, you'll see, instead of triangles, you'll see a triangle arranged as certain lines. What this means is that our repeat length is higher than the window size. So in this example, I think the repeat length is like 1,700 base pairs, and our window length is 1,000 base pairs. So because of that, it's going to be organized as lines. That's just an artifact of plotting, but still does the job. And alpha satellites, I think I forgot to mention in particular, are particularly interesting because they're form nested repeats. So a repeat of repeats, essentially. And so if you want to link different satellite classes together, you can look at rectangles that are orthogonal to the triangles on the diagonal. So if we want to see that the satellite between 0 and 1 megabase pair and 2 to like 2.3, 2.4 megabase pairs, we want to see they're the same, we can check orthogonally if there's a matching rectangle that fits the same identity profile, and there is. So in this case, we would say that these two satellites are the same class. We can say the same thing with like 2.6 to 3 megabase pairs, that these satellites are all HSAT3. And we can do the same thing with our alpha satellite. We see that they share a common rectangle orthogonal to it off the diagonal. So that means that they're the same class. So that was a pretty easy toy example. In reality, when you're dealing with large chromosomes, this can get significantly messier and a lot more harder to interpret. So this is a pretty large example here. This is the human chromosome Y. This is probably the most satellite-rich area of the human genome. And you can see that the bottom half of the chromosome is totally dominated by this, whatever this large satellite thing is. And this large satellite represents two alternating satellite classes. We know they're two alternating satellite classes because of all the gaps that are present within this big mosaic. So this just means that you have one satellite, next satellite, other satellite, next satellite. And this is verified by all of the off-diagonal rectangles that we see embedded within. We also have a bunch of duplications. So recall that duplications are not tandemly repeating, so they don't form these triangles, they just form lines. And they're pretty present throughout the entire chromosome, and they're pretty large as well. And you can see that they're embedded both outside of the satellite region, like at around 28 megabase pairs, and they're also embedded within the satellites. This is that difference between having a segmental duplication that's just a duplication versus having one that's also embedded within a satellite region. We also see some palindromes here, which is pretty cool. So palindromes are going to show up as perpendicular to the diagonal of your dot plot. This is just because they are inverted, and so anything inverted is going to show up as perpendicular. Okay, so that was self-identity plots. Next up is comparing two different sequences to each other to see how similar or how they share their satellite DNA. So if we look at these two sequences, then just by eyeballing it, you can kind of get a guess of what satellites are the same. We can see that whatever this sort of pinkish triangle is going to match with this pink triangle between sequence 1 and sequence 2. You can also guess that this heavy red triangle representing strong identity, this is probably a centromere or something, is going to match this strong red triangle between sequence 2. However, please do not compare plots like this. You will get fired if you do, literally. So if you want to compare two sequences together, then use the compare feature within ModDotPlot to produce a comparative plot. So this is the same thing, just sequence 1 is on the x-axis and sequence 2 is on the y-axis. Make this a little more clear. I'm going to arrange the data in a grid where we can see both the self-identity plots and the comparative plot arranged together. So the self-identity plots are going to be on the diagonal here. And the comparative plot, these two are the same. One is just transposed to the other, just to make it a little more obvious which is what. And if we want to see if they're the same satellite class, we can draw orthogonal lines. So that pink triangle that we were looking at is actually the same between sequence 1 and sequence 2. We can verify that because in the comparative plot, it exists. However, that big red block, when we take a look at the comparison here, nothing shows up. So what we can infer from that is that these are actually not the same satellite, even though just by eye, they have very high identity and they look similar. They're not the same satellite class. So very important not to assume that just because things are the same color, same identity, that they're going to be the same satellite. You need to look at the comparative plot in order to make that determination. We also see something quite interesting here. We see this large inversion. Recall that with palindromes, you have lines that are perpendicular to the diagonal. So this is what we see here. And this is that SST1 satellite region that I talked about with the Robertsonian translocations earlier in this talk. So this sequence here is chromosome 13, and this sequence here is chromosome 14. And what I've highlighted in the circle is that pseudo-homologous region that is the precursor for Robertsonian translocations. All right, so hopefully I've given you some confidence in how to interpret a dot plot. So now we'll go more to the interactive session of this tutorial, which is running ModDotPlot. So essentially, ModDotPlot can be run in one of two modes. The first is static mode. So static mode produces, like the name suggests, static image files. This is very lightweight. It's good if you know what you want and you want something that's publication quality and you want it fast. This is the route you want to do. However, ModDotPlot also has interactive mode. Interactive mode is built using Plotly and Dash, and it builds a plot hierarchy using that hierarchical modomizer structure that I talked about earlier. So with Plotly, what this does is launch an interactive viewing portal in your web browser. It's slower because you're producing multiple matrices using that hierarchy, but you can adjust your field of view in real time. You can play around with certain parameters like identity thresholds, colors. If you want to make a colorblind accessible figure, for example, you can do that. If you want to highlight different identity thresholds using non-traditional colors, you can do that. You can really customize the plot however you want, and then go into static mode and produce whatever you want. So let's start out with static mode. I'll switch out to the Gitpod in a little bit, but I just want to go over some of the basic parameters first. So the main parameters are going to be your input and output. Your input to static mode is going to be a sequence in FASTA format. This is pretty standard. When ModDotPlot computes sequence identity, it will produce a matrix in the form of a paired end bed file. So in the structure of this paired end bed file, you have sequence 1 with whatever intervals you're using, sequence 2 with whatever intervals you're using, and the identity estimate. So this is going to be the same regardless if you do a comparative plot versus a self-identity plot. And this is where the real computational time of the tool works, is taking this FASTA and producing it into a bed file. Once you have a bed file, then it will work to produce a dot plot, which is really more of a heat map when you think about it, because you're just coloring in areas of a matrix. And if you want, you can preload the FASTA files into bed files, store the bed files, and then when you want to produce plots in the future, you don't need to do the large computation of sequence identity. You can just directly go from the bed files to whatever plots that you desire. Some more parameters that you should be familiar with. An important one is the size of your sequence, because this is going to impact more downstream parameters. This is not something that you can control through ModDotPlot. If you want to narrow your field of view for static plots, you can use something like SAMTools FadeX in order to truncate your sequence or expand your sequence or whatever you want to do. So the main parameter is this R, or resolution. This is going to be the number of intervals that your plot contains. And the number of intervals that you have is going to be inversely proportional to the size of the window. You can think of the formula being n equals r divided by w. So if you want more windows, then your window size is going to be small. One important thing to keep in mind is that with ModDotPlot, your runtime is going to scale quadratically with the resolution, not with the sequence size. This is sort of the main difference between ModDotPlot and stained glass, where stained glass scales with the size of the genome. But aside from some overhead of counting k-mers, the runtime of ModDotPlot is really going to only matter with your resolution size. So just comparing how ModDotPlot works with different resolutions, we can see the parameters that are listed here. We have a sequence length of 6 megabase pairs, and we have a default plot resolution of 1,000. So do the math, 6 million divided by 1,000, we're going to get a window size of around 6,000. And this runs pretty quickly. You can produce a bed file of identity estimates in around 36 seconds. But now we can run that same sequence again, this time with double the plot resolution. So instead of segregating into 1,000 intervals, we're going to segregate into 2,000 intervals. And this ends up tripling the runtime, which makes sense because this does scale quadratically with resolution, but doesn't really have any impact with regards to the sequence length. If you used a very large sequence, it would also take longer when you up the resolution. So just keep in mind that runtime is dependent heavily on resolution, and that if you pick a good value with your resolution, you can get pretty ideal runtimes in every scenario. So just a comparison, these are two dot plots with two different resolutions. The one on the left uses 1,000, the one on the right uses 2,000. And you can see that they're pretty similar. Maybe the identity is more accurate in the 2,000, but regardless, you're going to get the same level of information containment. The main difference between using a higher resolution is in your histogram, where the histogram profile looks the same. But if you take a look at the number of estimates, you can see that it maxes out at 8,000 for your lower resolution, and for higher resolution, it maxes out at 16,000, indicating that you're using more windows to compare against. Okay, some other parameters to keep in mind. A big one is k-mer length. So ModDotPlot is a k-mer-based tool. Kind of the Achilles heel of k-mer-based tools is you always need to pick a k that's appropriate for your situation. If you use one that's too large, then you're going to be too specific, meaning that not every window has a k-mer of length 50. So you're going to lose out on a lot of data, and you're not going to get an accurate dot plot. On the other hand, if you use a k-mer length that's too small, you're going to be too sensitive. You're going to be having k-mers of length 6 in every window, potentially. So you're going to get an overloaded matrix. And so we set a k-mer length of 21. This is good for most situations. In some situations, if you want to be more specific, you can up it to 30, 31. If you want to be less specific, you can lower it down. But 21 tends to do pretty well. Now, if you want to do a comparative plot, you can use the compare flag. This will produce comparative plot if you have 2 or more FASTA or BET files input into your sequence. If you want to produce that nice looking grid I showed earlier, you can use the grid flag to do that as well. Although this tends to be a bit of information overload, so we limit it to 6 sequences if you're using the grid function. And this is what the command line will look like when you run on a pairwise sequences. You have sequence 1, which is 6 megabase pairs, sequence 2, which is 4 megabase pairs. Your window size is going to be based on whatever sequence is larger and produce a plot of the adequate resolution. Okay, so I think I talked enough about ModDotPlot parameters. I am going to go ahead and switch back to the to our Git pod. Oh, I timed out. Let's open that again. And we're going to go ahead and take a look at our sequences that we've been running. So I'm hoping that I don't have to recompute the static plots. If I do, it's not a big deal. It goes pretty fast.
Yeah.
Nice. Okay, it saved it, so I'm good. So essentially, let's take a look at the command that I ran. So I ran static mode with ModDotPlot. I ran it on a set of short arms, and I used the compare and the grid flag. So I did pairwise comparisons of all of those short arms and arranged it into a grid. And so this is what we get as output. We just get a bunch of plots. So we take a look. We can see that there were 3 sequences in our short arms folder. And we get a bunch of different files relating to those sequences. So we get a full sequence. This is the standard .plot output. Let me pull up an example. I think PDFs don't work with VS Code, so I'll just show the ping file here. It'll produce a PDF and ping of each plot that you produce. So this is one of the sequences. Looks good. We can see a big satellite class here, very small one here, big satellite here. Judging by the fact that the repeat length is larger than the window, this is likely RNA, rDNA. Then we have a big satellite class here. So this looks like it worked out pretty well. We also have the triangle. So this is the upper triangle portion of full. Really the same thing, just with the lower triangle removed, but has a bit of a shorter plot and looks a bit nicer if you want to overlay it vertically. Actually, this is quite nice. We can see here that there's a lot of duplications that are present in this satellite block. And those are those big lines that you can see within the satellite from 0 to 5. What's next? We have a histogram. So this is the sequence identity histogram. So the majority of them is going to be 100% identity estimate. This is going to be that diagonal. Anything off the diagonal is really what we care about. And we can see that we sort of have 2 peaks, one at around 90% identity, one at around 95% identity. We have comparison. So anything with the underscore compare is looking at 2 different sequences. And sometimes the naming of the sequences is going to get mixed up. Like here, for example, it's seq2, seq1. Doesn't matter. It's still going to be the same plot, either just transposed or not. Cool. So we can see that the comparative plot worked. We can see that rDNA is shared between the 2 sequences. We see that inversion we talked about earlier. So likely one of these 2 sequences is chromosome 14, and one of them is either 13 or 21, because we see that SST1 containing inversion in this plot here. What's next? We have the grid. So this is the same grid I showed earlier, except this time we have 3 sequences instead of 2. Hopefully this is somewhat visible to you guys. I will lower this a little bit. But we have the self-identity plots on the diagonal of this grid. So this is sequence 1, this is sequence 2, this is sequence 3. Another way to interpret this kind of grid style is you can think of the diagonal as just one huge concatenated sequence. And then anything off the diagonal is just the comparative differences between the two. So this actually is somewhat interesting. We have that same rDNA array. You can tell it's the same because orthogonally it shows up. Here we have this big red triangle that doesn't match up, but actually it does match up here, where you have this red triangle. It shows up much lower sequence identity. This is around 86 to 90% sequence identity to this satellite class here. So that's kind of interesting that these two match up. They are the same satellite class, likely, but show higher divergence compared to each other. You can tell they're more diverged just because of that sequence identity being reduced. So that's kind of cool. I think that's it in terms of sequences 1, 2, and 3. Yeah, so one of them contains an inversion. That would be sequence 2, because we see when we compare sequence 2 to 1 and to 3, we see that inverted palindrome there. Okay, so that was the acrocentric short arm part of the plots. Next, we're going to look at rDNA misassembly. So I mentioned that rDNA was one of the areas of CHM13, the new reference genome that was included. This is one of the more difficult regions of the human genome to assemble. It contains a lot of tandem repeats, and they span megabase pairs in length. So this command I will run right away. I won't preload it because it should go pretty quick. Oh, can't find ModDotPlot. Let's go. If this happens, you can go find the virtual environment, which I think might be in the folder here. Here we go. Now I want to paste. Retry. No, okay, I'll just copy this again. There we go, that one worked. So we can just wait around and see this in real time just because it's quite fast. So what ModDotPlot does first is it reads in a sequence and decomposes it to k-mers. It will take a k-mer and it will look also at its reverse complement. It'll hash them both and then pick whatever one is lower in terms of their integer value. So if you don't do this, if you don't look at the reverse complement, then things like palindromes or inversions aren't going to be visible in ModDotPlot because you're not taking that sort of inverted characteristic into account. Ooh, I got the wrong folder, so I'm going to switch my command to, I'll just do it by hand because it's faster. We want rDNA. So we want to find our FASTA files in the rDNA folder. It's going to be in BGA data. rDNA, we want all.fasta. You can use wildcards when you're selecting inputs for ModDotPlot, which is handy. We want to save that in output/rDNA. We want to compare, and we want to produce a grid. Oh, I forgot to select that we want static. There we go. So collecting the k-mers for this is super fast because our rDNA length in both of these instances is around 1 megabase. And so you can see with the math here that for a sequence length of around 1 million, we have a window size of 1000 because our plot resolution is going to be 1000. So math checks out there. You don't have to worry about this monomizer sketch size, but the sketch size was that sampling that I talked about earlier, where you can downsample the amount of k-mers that you select by taking a different sketch size. This plays into account when you're working with larger sequences like entire chromosomes, then you're going to sketch a lot of your window compared to these small sequences. We're not going to be doing any sketching at all. All right, so I think one of the rDNAs has finished, so we can go ahead and take a look at that. That's going to be in our output, rDNA hap1. So this is one of two different haplotypes. I'm not seeing the plot. I don't know if my, oh, there it is. Just took a little bit to render. And this is nice. This is what rDNA is supposed to look like. We can see a bunch of different lines off the diagonal. This represents the rDNA copy number. So if you go ahead and tediously count all of the lines that are present here, it's like 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So this is a 16 copy number rDNA. Cool. This is probably from chromosome 14. I think that one has the lowest number of rDNA copies. And this ranges, by the way, from I think 14 to 71. I think chromosome 15 is crazy with how much rDNA it has. So with the 71 copy rDNA, it's not really realistic to like count all the copies out, but you can do that if you are so inclined. I think the second haplotype is done, so I'm going to go ahead and show that. And you can see that this one is very different from the first one, where we actually have a satellite in our rDNA here. You can tell it's a satellite because it shows up as a triangle on the diagonal. Not really a triangle, but a square in the full dot plot. If I was to open up the tri version, which is here, then I'll see a triangle, which I do. And as a result, what happens with these tandem repeats is that they make a bridge. So anytime you see anything mismatched in terms of lines and stuff, that's indicative of an assembly error. So this is likely what happened with this haplotype, is that this was just assembled incorrectly. You have this satellite kind of messing things up. Likely this isn't a real satellite. Likely it's just a repeat region that in your assembly graph, whoever assembled this, probably not whoever assembled this, whatever the auto assembly method was, just assumed it was the same sequence repeating over and over and over again. And that's what leads to this satellite class here. So yeah, this is an assembly error. We can see that haplotype 1 doesn't have that, but haplotype 2 does. And if you want to make it even more clear, we can go to the grid, take a little minute to load, and see that this is our correct haplotype. This is our misassembly, and that in a comparative plot, we see this misassembly show up, not as a satellite, but as mismatched arrays. Okay, so that was rDNA. I have around 10 minutes left, so I think I'm going to go over some interactive mode commands. So one thing with interactive mode is that it launches a Dash application that requires local host access. So normally if you're on like a high performance computing environment, that's an issue because you don't have access to local host on that. Local host requires you to be on your computer. VS Code actually has a super awesome feature called port forwarding that's built into it. So with port forwarding, you can go to this ports tab and you can see that it has two open ports. One is 5900, one is 6080. This allows me to view stuff on the web on my own personal web browser that is being hosted by the server, which is Gitpod in this case. And so we can do this with interactive mode as well. We can run ModDotPlot in interactive mode, and we can launch a port at port 8050. And what this will do is forward that port to our local host so that when we go to our personal web browser and go localhost 8050, we can see whatever showed up on 8050 on the server. So this is really cool. This allows you to use interactive mode on a computing cluster. So let's finish up the command. I preloaded a bunch of Arabidopsis centromeres. So Arabidopsis is that plant that ModDotPlot could compute the matrix for in 5 minutes. That was in static mode, and that was with a high window or a low resolution. With interactive mode, because we're dealing with a lot of high resolution plots, this is going to take much longer. So to save some time, I preloaded it. And let's actually remove this and paste the command. So I preloaded these matrices. ModDotPlot's going to look for the folder that they're in, read, scan the data, and then launch an instance of the local host. So one thing to expect with port forwarding is that depending on your server, it can be high latency. So ideally, you want to run this on your personal machine because there's going to be no latency there. Obviously, if you're dealing with a lot of sequencing data, you don't want to port that to your local machine. So sometimes you have to use your HPC environment, but just keep in mind that server latency can sometimes be an issue. I might switch in a minute to VS Code on my local machine if this takes a while, just because I don't think I requested enough memory in this Gitpod instance in order to launch it. But okay, now it works. So that's good. So you can see it'll say ModDotPlot is running interactive mode on localhost. This is that 127.0.0.1 and port number 8050. And if we go to the port tab, we can see that a new port has opened up at 8050. So if we go there, let's just click this, see if it works. Yes. Take a minute to load, but there we go. We got interactive mode. Woohoo. So I'm going to close this and relaunch this in my personal instance of VS Code, just because with the latency on Gitpod, it's just going to take a little while. Let me share my VS Code. I'll just share my desktop. There we go. So this is running on my personal machine, and this is running on port 8050. And you can see that when I run it, not on Gitpod, but on my local machine, that it does a much better job at loading. So this is a Arabidopsis centromere. So we can see that we have two parameters here, a current window size and a minimum window size. So the current window size is what we're looking at now. If we zoom in, we can zoom in just by dragging. Then it will take a second to re-render, and then it'll switch. Our window size went from 1600 to 16,000 to 8,000. So every time we zoom in, we get a little bit higher resolution plot. And so we can do it again. Boom, now we're at 4,000. We can do it probably one more time. Yes, there we go. So now we're at 2,000. This is our maximum resolution. We can't zoom in anymore because we preloaded 4 matrices, but these 4 matrices are good enough. They do the job. And so now we can just play around and have some fun. So you want to update the matrix to maybe mask lower identity values. You can do that with this slider. Play around. If you only want very high identity regions, you can do that. If you, for example, want to produce a colorblind accessible plot, we have a bunch of different palettes that you can play with. Orange to red, oranges has been shown to be quite popular. If you also want a more like qualitative color pattern, like for example, you want to really highlight the difference between 98% identity versus 97% identity, you can use one of these, even though they look kind of ugly. It really is a stark contrast in terms of different identity thresholds. So you can pick whatever you want. You can reset just by clicking this home button here. And with ModDotPlot in interactive mode, you can load up to 2 matrices and 1 comparative plot. So this is chromosome 1 of the Arabidopsis. It's around 32 megabases. Move on to chromosome 2. This one's smaller. This one's around 24 megabases. And its centromere is in a different location. It's at around between 3 to 8 megabase pairs versus the other one, which was at around 20 megabase pairs. We can do the same thing. We can take a look, wait for it a second, it'll re-render, and you get a new image. Cool. Move back. Now we can see the comparative plots, see how their centromeres compare to each other. So this is the comparative plot of their centromeres. We see that the centromeres do share identity around the 96 to 90, maybe 90% range. So that's kind of cool. You can see that those centromere satellites are somewhat conserved. And the neat thing about this is when you find a plot style that you like, so for example, say that I want this centromere, but I want it maybe only from 94% identity and higher. I don't care about the other stuff. Then you can select that and then save matrix to bed file. And so what that'll do is you save your bed file to your folder. If we go to my VS Code here, it'll say save chromosome1.bed in my folder here. And this is really nice because I can, where'd my ModDotPlot go? There it is. This is really nice because if I want to produce a static plot in the future, then I can just take that bed file, run ModDotPlot static on that bed file, and get the result. So yeah, that's interactive mode. I have one more thing, but I think I'm out of time, so I won't go over it. But if you want, you can compare a set of chromosome X centromeres to see how well they stack up. This is kind of a fun little comparison you can do, and we use the result of this comparison in the ModDotPlot paper. So to wrap up, I will go to our slide here. This is parameters, and I want to thank a bunch of people, mostly my lab at the NHGRI, headed by Adam Philippi, Sergey Koren, and all of the others, as well as my lab at Johns Hopkins, headed by Michael Schatz. Both Mike and Adam have been amazing advisors and have really spearheaded this project on looking for repeats. And thank you, Damon, for all of your work hosting BGA. So that's it. If you have any questions, feel free to reach out to me afterwards or let me know on the Discord or anything if you have questions. Thanks.
Thank you very much. I have a quick question while you're still live, if that's okay.
Yeah, of course.
So is there identity levels that's customizable? So I could have as low as identity level as I want on the identity plot. Is that a fixed thing?
That is a parameter. I didn't go over it, but yes, you can set the identity parameter to be whatever you want. Ideally, we'd recommend you to not go lower than 86%, just because the way we compute identity really decreases in accuracy if you go below 86. Like if you want to do 50% identity, then the results aren't going to be very meaningful.
Yeah.
But yes,
okay.
You can customize it to however you want.
Fair enough. And so surely there's a lot of use cases for this. I mean, even the underlying minimizer and sketching for this there's a lot of use cases. So is there any plans on expanding it so that it could take on something like high glass, for instance?
Yeah, I think high glass uses the cooler format. This is, I think, developed initially for high C data, but this is kind of the same thing where you're just storing a matrix. So yeah, integrating that hasn't been on my radar in the last little bit, but it's definitely been a feature people have been asking for. So I'm looking into adding that functionality as well. There's sort of two main expansions that I want to do with ModDotPlot, and hopefully I'll get to them later on in my PhD. The first one is having annotations be automatic. So essentially, given a dot plot and some machine learning type capabilities, give me an annotation. And once that annotation's done, display it underneath the dot plot in sort of an IGV style viewer.
That sounds very cool. Thank you very much. But yeah, I would be very interested in testing out a high class competitor. Anything that can take less than overnight to run is a godsend in my book.
Mm-hmm.
If does anyone else have any questions? Deafening silence. Yeah, if there's nothing else, then again,
I have a question.
Okay.
Go.
Is there a size limit on the chromosomes that you could visualize?
Good question. There is not. You can do whatever you want practically. There is a number limit in interactive mode. You're limited to 2 sequences and a comparative plot. You can't do multiple sequences. Also, if you're in that grid visualization, you are limited to 6 sequences. But in terms of the size of the sequence, go nuts. You can do entire chromosomes, you can do entire genomes if they're concatenated together. Your only issue is going to be memory. If you use entire genomes, you might hit a memory wall. So, but ModDotPlot allows for arbitrary length input.
Thank you.
Right. Anyone else, or should we let Alex get on with the rest of his day? No? Okay. Fantastic. Again, thank you very much, Alex, for giving up your time to run the session. And I hope to see the rest of you in another session. Thank you very much.
Right. Thank you.